Good morning and warm welcome from Indonesia to our honorable speaker and moderator of today's international guest lecture. Lecturers, students, and participants, before we start the opening ceremony, please allow me to introduce myself. I'm Agnes Anindita as today's Master of Ceremony. We all are here today with the spirit of the 61st anniversary of the Faculty of Farmasi Universitas Pancasila. Today is such a great opportunity for us to have another series of international guest lecture with the topic of the role of pharmacogenomics in precision health. Ladies and gentlemen, before we move to the next agenda, we would like to invite you to sing the national anthem of Indonesia, Indonesia Raya, followed by the hymn of Universitas Pancasila. For all participants, please be seated properly. Thank you.
Tetapkan hati masa depanmu. diberlakukan pada dasarnya dan mesti dijaga terus.
Ladies and gentlemen, let's move to the first agenda. The first agenda is the opening remark, which will be delivered by the Dean of the Faculty of Pharmacy Universitas Pancasila, Professor Dr. Apoteker Syamsuddin, Master of Biomed.
Thank you, Agnes. Good morning, everyone. Assalamualaikum warahmatullahi wabarakatuh. The Honorable Associate Professor Dr. Beben Binyamin as speaker, the Honorable lecturer, student, and participants. Professionally, as Dean of Faculty of Farmasi Universitas Pancasila, I express my high appreciation for the presence of Associate Professor Dr. Beben Binyamin from the University of South Australia as a speaker at the guest lecture. The topic of this activity is the role of pharmacogenomics in precision health, which is important in providing broader insight and knowledge for students, especially increase their knowledge of molecular pharmacology. As we all know, there is involvement of gene and protein in the course of disease and the body's response to drugs. And pharmacogenomics is a field of science which is believed to be able to explain that difference in the response of each individual to the drug given are closely related to the genetic difference of each individual. So, the topic of the role of pharmacogenomics in precision health is important for us to know. This guest lecture activity is one of series of activity in the framework of the 61st Faculty of Farmasi Universitas Pancasila anniversary. I hope that the vision and participation of all participants can take advantage of this moment to continue to gain knowledge from the speaker. Finally, we hope that this guest lecture can provide new insight, inspiration, and deep understanding for all of us. Thank you very much.
Thank you, Professor Syamsuddin, for the wonderful remark. Now we are on to the next agenda, which is the photo session. For all participants, please turn on your camera. Okay, let's begin on the first slide. On the count of 3, 1, 2, 3. Now we're on the last slide. On the count of 3, 1, 2, 3. Thank you. Now let's move to today's main event. Pharmacogenomics is the study of the role of the genome in drug response, which investigates how a person's genetic information influences the reaction to drugs. By understanding how individual genes affect medication effectiveness, we can customize drug treatments to each patient's need, maximizing therapeutic benefits and also minimizing the risk of side effects. It's such an interesting topic to discuss because pharmacogenomics is revolutionizing medicine, paving the way for a future of precision medicine where treatments are designed specifically for each patient. Before we begin the lecture, Please allow me to introduce our moderator, Apotheker M. Iqbal Julian, RPP, PhD. Mr. Iqbal is currently a lecturer at the Fakulty of Farmasi Universitas Pancasila. He has several training experiences, including basic ethical review and good clinical practice. He is also an active researcher. Some of his publications are investigation of the anti-hyperuricemic effect of Gandarusa leaves ethanolic extract in hyperuricemic rats, and investigation of biomarkers and handling strategy of erlotinib-induced skin rash in rats. Mr. Iqbal is already with us now. Good morning, Mr. Iqbal.
Good morning, Miss Agnes.
Okay, so without any further ado, the lecture session will be started. Mr. Iqbal, the time is yours.
Thank you, Miss Agnes. Good morning to lecturers, students, and all participants. As introduced by the MC, Miss Agnes, my name is Iqbal, and I am the moderator for the International Guest Lecture 6 series. Today, we will learn pharmacogenomics, specifically the role of pharmacogenomics in precision health. Hopefully, from this lecture, we could gain insight on what is pharmacogenomics, what is precision health, and what is the role of pharmacogenomics in precision health. The lecture itself will be presented by Associate Professor Beben Benjamin. Good morning, sir. Okay, nice to meet you. Okay, first, let me introduce our speaker. Associate Professor Beben Benjamin is the group leader at the Australian Centre for Precision Health and research degree coordinator at the Allied Health and Human Performance Unit, University of South Australia. He graduated from Bogor Agricultural Institute, or IPB University nowadays, for bachelor's degree in year 2000. Then the University of Sydney, Australia, for master's degree in agriculture in year 2002. And the University of Edinburgh, United Kingdom, for doctoral degree in year 2007. He did his postdoctoral training at the Genetic Epidemiology Group, QIMR Berghofer Medical Research Institute, from the year 2007 until 2011. His current interests are in the application of statistical genomic methods to neurodegenerative and psychiatric diseases, such as motor neuron disease and schizophrenia. Associate Professor Beben Benjamin has supervised and mentored research students and is currently available to supervise new research students to study in the exciting areas of applied statistical genomics, genetic epidemiology, big health data, and genomic of complex diseases. This is an interesting point for lecturers or students who study desires met Dr. Beben expertise. Is it correct, sir? So we can apply for continuation of our study. Okay, that is our speaker introduction, and we can argue that Dr. Beben Benjamin expert today's lecture topic. Before the presentation begins, these are the instructions of this lecture. The lecture will be presented for maximum 40 minutes. After the lecture, the question and answer session will be open for 20 minutes. During the Q&A session, participants can ask questions directly to Dr. Beben or indirectly through the chat column. Okay, those are the instructions. Next, let us begin the main agenda of this event. Dr. Beben, if you are ready, you may begin your presentation.
Thank you very much, Dr. Iqbal. First, I would like to say happy birthday to Faculty of Pharmacy for your 61st anniversary. I think that is great achievement. Yeah, congratulations. Thank you for Pak Dekan, Professor Samsudin, for inviting me to give a lecture here at the Faculty of Pharmacy. I'm delighted to share some of some of our work, including also some overview of the pharmacogenomic development at the moment. So I'm going to share my screen. Can you see my screen okay?
Yes, sir, clearly.
Yeah. Good. Thank you. So today I'm going to start from the very basic about human genetics, and then understanding the role of genetic in human disease, and then how do we find a gene associated with disease, and then how do we use this information in precision medicine. And then I'll give you some of the example of development in genetic studies to identify drug response or pharmacogenomics. Okay, first, I think it's important to understand the difference between what we call as a Mendelian disease and complex disease. So from a genetic perspective, all human condition or human disease, or including human characteristic and trait, they are influenced by genetic, and then also some influenced by environmental factor. We sometimes call it a genetic disease or penyakit genetic, and the other is penyakit or disease that caused by environment. But in fact, from genetic perspective, all of disease are influenced by genetics. Yeah? The only difference is that Mendelian disease, or what we call as genetic disease on the lab, is that the disease is influenced by the mutation in single gene. Yeah. For example, cystic fibrosis, thalassemia, probably the most common in Indonesia. They are influenced, or sickle cell disease influenced by the mutation in single gene, and then the effect is so large that if you have that mutation, and then you are almost the minute that you get the disease. But it's also sometimes it's not fully penetrant, influenced by other modifier disease, and then influenced slightly by the environment. So on the other hand, so this is what we call as Mendelian disease: cystic fibrosis, Huntington disease, thalassemia, sickle cell disease. So this is that what we call as penyakit genetic. Yeah. But in fact, all other disease that we experience, for example, type 2 diabetes, cardiovascular disease, asthma, schizophrenia, and everything, they are also influenced by genetic factor and environment. The only difference is the genetic pattern of inheritance, how it's inherited from the parent to the offspring, are not clear. Because it's influenced by 100 mutation across the genome, which each has having a small effect, and then also influenced by environmental as well, and interaction between the genetic and environment. Yeah, so I think that's important to have this understanding of the distinction. We as a geneticist are trying to understand that genetic contribute to disease, and then also we want to find out which actual mutation in the genome that affecting disease. Okay, so these are some of the historical method applied to a complex disease. Yeah. With a single gene mutation, it is it's much easier to find the gene, the actual mutation, because you can just follow the inheritance of the family pattern, large pedigree, for example. You'll be able to find that that this disease is co-inherited with the inheritance of genetic marker. But for complex disease, it's quite different, quite much more difficult because the effect is very small. So first, in the early 80s, there are a lot, until early 80s, there are a lot of studies, twin and family studies. So it's basically, the typical study is by comparing the similarity or the resemblance between family members, that we will be able to say like how much relative contribution of gene or genetic compared to environment in influencing disease. Let's say we'll be able to say that the difference between individual in their height or stature, that's 80% of them influenced by genetic factor and 20% by environmental factor. That is from twin and family studies, yeah, by comparing the resemblance. But that doesn't tell us what specific gene or where are the gene in the chromosome. Okay, so it's only tell us that this particular phenotype, this particular disease or characteristic are to some degree influenced by genetic factor. Okay, so we've done a number of different studies to say that most, like our review in the last 50 years found that most of the human characteristic, or almost every human characteristic, a human disease, are somewhat influenced by genetic factor. The only difference is by how much. Then in the until early 90s, linkage studies by following the pattern of inheritance between family member will be able to find gene mostly associated with Mendelian disease or simple disease or penyakit genetik, yeah. So be able to identify that this mutation in, for example, CTFR gene are associated with cystic fibrosis, for example. And then candidate gene studies where individual, and then linkage study was not successful in complex disease. So it means that people now move on to candidate gene studies for complex disease. It means that we focus on one gene at a time, and then look for the association of the mutation within the gene with disease. But then it also has a problematic in the sense that a lot of false positive, a lot of association that are not real been identified. So there are hundred or a thousand of literature where the content are, that most of the association are false. Yeah, it's not correct. Okay, so that's why in human genetic we kind of abandon candidate gene studies. It's difficult to publish any candidate gene studies nowadays. So because it's found that it's to be unreliable and then create a lot of false positive. Then we move on to what we call as genome-wide association studies for the last 15 years or so. Basically, instead of focusing on a single gene within a gene, a single mutation or a group of mutation in a gene, and now forget about a gene. We want to identify everything, the whole genome. Like we try to find association in every part of the genome. So they are called genome wide. So it means that we don't focus on a single gene, but everything. And then also can be done using a sequencing study. And then with each studies, the sample size requirement also increase. Yeah. So it means that larger study are needed, larger number of individuals are needed. Okay, so how genome-wide association works? So genome-wide association, it is not a new method. So it's been around more than 15 years, but it is the most successful gene finding method in human genetics. So what genome-wide association study is that we collect hundred or thousand of individual who has disease, compared to individual who does not have disease or healthy individual. Let's say you compare, you collect a group of individual who has cardiovascular disease compared to healthy individual. We collect all the clinical information on the individual, and then also we collect genetic information of individual, genotype them, and then we compare whether there is any differences in genetic that are associated with a disease. Yeah, so compare just doing association studies between the genetic difference with the phenotypic difference. And then because you are analyzing a million of genetic marker or change in human DNA base pair or single nucleotide polymorphism. So what you come up is what we call as a Manhattan plot like this. So it means that each dot is an association between single genetic mutation with a disease. You can see here that the one that so for example, this is an example of a gene for iron that we found several genetic mutation associated with the iron level, yeah, in the body. Chromosome 3, so chromosome 2, chromosome 6, chromosome 9, and so on. So this is another example. So this is a genome-wide association result for motor neuron disease. So one of the disease that we studied, basically, we found, like for example, this gene GPX3TNIP1, and then these are some of the. If we zoom in of this location, we can see that there are a number of different single nucleotide polymorphism that are associated with motor neuron disease. Okay, so we can see that if you see the x-axis here, it's a number of chromosomes, and the y-axis here is the minus log 10 p-value. Minus log 10 p-value is just the way we present them. So it's basically minus log 10 p-value of 10, it means that the association p-value of 0.00, you have 10 zeros, yeah, 1. Instead of standard p-value of 0.05, which no longer applicable here because you are doing a million of testing, so you accept that any SNP associated with this disease is significant if the p-value is more than 5 to 10 to the minus 8, yeah, rather than 0.05. So that's why we call it as a genome-wide association significant threshold. Okay, so the line here. So because if you consider 0.05, there will be hundreds of thousands of SNP that are associated, which are the result of multiple testing issue. And then we are not interested on that. Okay, so that's different between GWAS and the standard association analysis. And then you can see here that there are a progress in the number of publication for GWAS. So you can see in the blue, so starting from 2007 to 2022, for example, you see the increase in the number of publication to almost 140, number of publication in the order of the blue one here. So it's more than 500 per year, for example. And then average number of SNP or gene location that are associated with disease in the green here. So you can see that there are identified new mutation or genetic variation, about 40 per year, and the number of publication for also the red ones, so number of sample increase as well in the order of 100,000, 150,000. Yeah, so it's a big number, required big number because the effect size is very small. All right, now, like how much this genome-wide association has been done in pharmacogenomics, right? You can see here that the number of publication in pharmacogenomic is also increasing, but it's quite stagnant after 2016, for example, but probably on average about 40 publication per year. But I need to update this number for the last 3 years or so. But you can see here, a dot here, the number of proportion of pharmacogenomic related genome-wide association studies compared to the overall genome-wide association studies for disease or trait. You can see here that the average number is about 10% or so. Oh, yeah, between 5 to 10% of all genome-wide association studies are performed or conducted for pharmacogenomic related trait. And then you can see here the number here. Yep, some sample size as well is usually much smaller in pharmacogenomic compared to all other GWAS. So these are some of the example of the finding from the pharmacogenomic genome-wide association study. You can see, so I'll give you, I'll point to one of the example here, the dot here, yeah. So this blue dot here, you can see the lab is on warfarin. So this is, I think you already know, it's a blood thinning medication. And then if you go down, there is a mutation within this, yeah, CYP2A6 that are associated with how people respond to this medication. I think so. I think one of the response is that it took long to metabolize the medication. And then another good example here as well, it's ribavirin, which is drug for, one of them is for treating hepatitis C. The response of individual, it's also the influenced by the mutation within ITPA gene. Yeah. So if you can have a look, this that red one is adverse drug reaction. The yellow is biomarker, green metabolism, and then blue are response. And then so these are some of the example that you can see. Okay. So what is the challenge? The challenge at the moment with the genome-wide association studies or genomic in general is that how transferable are these findings? Because most of the findings are from the European, how this can be translated directly or easily to other populations such as Indonesian population, for example. You can see that I think the figure is still relevant until today, that around 80% of all the genome-wide association studies are done in European population, and then only less than 20% in other population. And then we know that genetic frequency, because of the population history and then also evolution, can be different between population. For example, the lactose intolerant gene, LCT gene, they influence whether an individual be able to digest lactose, which is a component in milk, into sugar, and it's influenced by the mutation. It means that people who does not have the mutation in LCT gene, if they drink milk, they're more likely to have a reaction such as bloating or diarrhea, which is, the frequency is very high in Asian population and can be as high as 60%. So that's why I think it is quite common for Indonesian, including myself as well, that we could not digest milk unless it's been processed into, for example, yogurt or other processing. For example, I usually drink lactose-free milk where the lactose component of the milk has been digested into sugar component, which is galactosa and glucose. Yeah, if I'm not, yeah. And then also the other thing is the strength of the association can be different between population. For example, the MHC gene that affecting schizophrenia, different association strength in European and Asian population. So that's why, although the finding has been found, the finding probably applicable in some population might not be applicable in other population. So we know that South Asian population, which include Indonesia, home to 500 million people, is not really well represented in this GWAS research. It's only a small number of GWAS research from Indonesia. The other thing is that relevant to pharmacogenomic as well is that different drug response can be different between ethnic group. This difference can be, at some respect, is influenced by genetic difference. One of the known example is carbamazepine, which is drug for treating epilepsy. It is for some people who carry this type of mutation within the LGLE gene, it might lead to adverse reaction, which is kind of drug reaction, which is called Stevens-Johnson syndrome. And then the interesting thing, or unfortunately, that this mutation is quite common in Asian population, but it is very rare in, for example, in a European population. And then there are some other examples as well. So it is important for some drug, for example, that understanding that might have a different response in different population, which means that it is important before giving this medication, for example, to check for some of this a genotype of that individual to avoid the adverse drug reaction. So what did we do to answer the challenge that genome-wide association studies are mostly in European population? So we did genome-wide association studies, for example, by combining European population and Chinese population. And then we found this new gene, that GPX3-TNIP1, for example. And then also our student as well, it's developing method to use genetic data to estimate the correlation between different ethnic group, for example, for some trait or disease. And then the other thing that it is important to understand is how all of this finding related to this, what we call as precision medicine or personalized medicine or precision health. So currently we know that if a patient come to a clinic and then they complain about high cholesterol level, and then most of the medication at the moment is statin to reduce the cholesterol, and then work for most of the patient, average patient, but not for all. Probably for most, it improved the outcome where the cholesterol level decrease, but for some patient, it provide no benefit at all. And then even for small proportion of patient, it might lead to adverse side effects such as muscle pain, for example. So what we want, I think, is that we know that this different response of people due to medication are partly influenced by the genetic mutation, but that's not all. I mean, it's also influenced by environment and then also by other things. So what we want is we will be able to use genetic and other information to be able to stratify individual. Okay, instead of giving the same medication to everyone, we want to be able to group individual into one or two group or three group that, okay, so it's mean that based on the genetic profile that, oh, okay, this group of individual most likely to respond well to statin. And then this group of individual might not have desired effect. So it means it's drug does not have effect. And then the other one is probably people who will develop or might develop side effects. So we only given that medication to people in blue here who might improve the outcome, and then probably look for an alternative medication for people that group in yellow and red here. Yeah. So it means that it's better for efficiency of drug use and then also better for patient. Okay, so how do we create this kind of one of the information used is by creating what we call as polygenic risk score. So polygenic risk score is basically combining all the genetic effect of SNP effect of a single individual, because like one single nucleotide polymorphism or one genetic mutation only affect the effect is very small to the disease. Yeah, this is an example. Let's say that from genome-wide association studies, we found, let's say, how many gene here? Okay. Yeah, so we found like 4 genetic mutation or single nucleotide polymorphism, that SNP1, SNP2, SNP3, SNP4. And then you know that if individual has an A allele, they increase the, let's say, the blood pressure in term of unit of let's say a systolic blood pressure increase by 1.5 point. If you have C, decrease by 0.5 point. If you have T, increase by 2 point. If you have A, minus 1.5. Okay, so that's for example. And then we know that there are 100 of this SNP affecting blood pressure. So you have 4 individual here. So each individual have a different genotype for this SNP. For example, you have AT for SNP1, CG, TT, and CC for SNP4. And similarly for the other individual. So a polygenic risk score is calculated as follows. So because we know that for SNP1, A has an effect of 1.5, so you put 1.5 here. C is minus 0.5 because we know that individual has C, so it's minus 0.5. T2, we know that there are 2 T here. Each T has increased blood pressure by 2 point. So you have 2 by 2 is equal 4. And then A is minus 1.5. Because this individual does not have A allele, it's only CC, that it means 0. Okay, so you average them out and then you get 5 here. 1.5 minus 0.5 plus 4 is equal 5. And then you do that for all individual you have and then put them in the distribution of polygenic risk score. You can see that individual 1 here among is individual who has the more high risk of developing a blood pressure with the score of 5 compared to individual 4, which are less likely to develop high blood pressure because score of minus 5. Yeah, so that is a simplified explanation of how polygenic risk score is calculated. So we applied this with to polygenic risk score to, for example, to predict individual likelihood to develop motor neuron disease. And also we also applied this how polygenic risk score, if we combine with lifestyle score, whether they influence the outcome of cancer. We know that cancer are influenced by genetic and environmental factor, and then we know that healthy lifestyle might reduce the risk of cancer, but we want to know whether individual who has high risk genetically for cancer might be offset or might be better if they adhere to healthy lifestyle. And then we found that for most of the cancer, healthy lifestyle can offset the genetic risk. Yeah, but basically, regardless of your genetic risk, healthy lifestyle might improve the outcome of cancer. Yeah. Okay, so another challenge is that the genetic risk score is not translated well across different ancestry. You can see here that the precision is not good, which you can see that the higher or a wider confidence interval here. So if you develop polygenetic risk score from GWAS population in European applied to European, it's quite precise. But if you apply to other population, it's not that precise. The prediction accuracy decreased from 1 to 0.5 on average. Yep. That's why research in developing country and different population than European one are important. So this is another work by Momin, our student on the cross-ancestry genetic understanding different ancestry genetic architecture between different tribes. So in summary, I hope I'm good with the time here that genome-wide association study have found that there are thousands of genetic variant or genetic mutation that are associated with hundreds of thousands of human disease. The GWAS and pharmacogenomic trait contribute only to less than 10% of all GWAS in human. And then the challenge in both GWAS or in GWAS pharmacogenomic is that most of the study is from European ancestries and the transferability of other population is currently being investigated. The genomic prediction, a prediction of individual risk to disease based on the genetic information, has promising clinical application, but the transferability between different countries or between ancestries is not good as within ancestry. And then this is the basis of what we call as personalized medicine or precision medicine or precision health. So within our center, so we have a range of different research, not only in genomic here, which is my research, and then also in population health, for example, epidemiological studies, analyzing large data in epidemiology, and then also a translation, trying to translate this finding into clinic. And then this is my kind of place to work here, this building here. And then these are outlet from above. And then with this, I would like to thanks our collaborator and student for the work that I presented here today. And then also thank you very much for Faculty of Pharmacy, Professor Samsudin and Dr. Iqbal for inviting me to give a lecture here today. I'm happy to take any question. Thank you very much.
Yeah, thank you for your insightful presentation, Dr. Beben. Okay, next we move to the Q&A session. For the participants who want to ask questions directly, please click the raise hand button, then inform us your full name followed by the questions. As for the participant who want to ask question indirectly, please type your questions in the chat column. Okay. Yes, sir, while we are waiting for the questions from participants, I have a question to ask. Prior your lecture, I thought that the terminology of pharmacogenomic overlap with the term pharmacogenetic.
Yeah.
Could you explain what is the difference? Is it based on the classification of disease, as you mentioned in the earlier slide,
Yeah.
Mendelian disease and complex disease, or what?
Okay, that's an interesting question. So there is no such very kind of strict definition between the two. Sometimes people use interchangeably. But I think the most accepted differences between the two is when you talk about genetic, is we talk about studying a single gene at one time, focusing on single gene and how that gene inherit what the pattern of inheritance of that gene within between generation of families. Yeah. Genomic is more on not focusing on single gene but the whole genome thing approach. So yeah, I think that's where you focus on all of the gene in the genome. So that's why I think the distinction between genetic and genomic are made. Yeah.
Okay. Okay, thank you, sir. Is there already questions in the chat column? Okay, it's not yet. So I will ask another question, sir. Actually, the classification of disease kind of new insight for me, that there is a Mendelian disease and a complex disease.
Yeah.
Now I want to move to the research of the GWAS, genetic-wide association studies. How can we justify when we compare population with disease and population without disease? How can we justify that the people without condition or without disease don't have genes responsible for the disease?
Yeah, that's interesting question. So first on the so I think so that why it's in the early slide. So I tried to give briefly description about that from genetic perspective and like kind of the all disease or human condition influenced by genetics. And the difference is in the pattern of inheritance, a different pattern of inheritance, because I think from most people understanding that, okay, so you can classify this is genetic disease and then this is not genetic disease, something like that. But that's not true. So on GWAS, so when you collect data on, let's say, 1,000 individual with a cardiovascular disease, and then 100,000 of people without cardiovascular disease, and then also you take the genotype. Okay, so you type the genotype using let's say Illumina array. Like, so you can for each individual you will know the genotype of each individual for each million position. So you're correct that all of the individual, whether you have condition or you don't have condition, you will have the same like a gene, like the same gene. The only difference is that the variation. Let's say, for example, so you compare the frequency. For example, for SNP A, okay, or genetic variant A, that you have the allele. So you have two gene A and T, for example. Yeah. So you calculate that in people who has disease, the A frequency is, let's say, 80%, and then the T is only 20%. Yeah. But in normal individual or people who does not have disease, the frequency of A and T are different. Let's say the A is only 40%, and then T is 60%. So that difference, if you calculate, like analyze statistically, that's what constitute whether that particular SNP is associated with disease. So basically comparing the frequency of the mutation between group of individual who has disease and a group of individual who does not have disease yet.
And then we can get the risk score.
Risk score, yes, that's
Yes.
correct. Yeah. Based on comparing the frequency, yeah. Risk score of each of the allele, yeah.
Okay. Yeah, understood, sir. Thank you. Was there any questions? Okay, there's no. Okay, sir. I would like to ask another question, sir.
Yeah, sure.
I'm interested in this study actually, but don't have any background on this genome study. What your suggestion to students who have passion in genomic study? What should they do first, and the other steps? I think that's the question, sir.
So the question is,
Please-
I
please suggest.
understood correctly, is like what step need to be taken if student is interested in doing
Yes.
genomic research, do you mean?
Yes.
Okay.
Correct, sir.
So there are range of different type of work. I think in genomics. So in my case, or in our lab, in my lab, is that we call it as a genomic of complex disease. So we are trying to understand the genomic of complex disease, which is some of the research that I presented. So first, student need to be interested in numbers, because we don't do the research in wet lab, but we are doing the research in dry lab. So we use computer. Basically, we collaborate with other researcher who produce or has the data, with clinician, with doctors, and then also molecular biologists, and then us is analyzing the data to make sense what the data tell us. Yeah. And then also we use a publicly available data as well, in publicly available genomic data that we can access. So it's mean that first student need to be interested in numbers,
Statistics.
don't afraid with using computer, that they need to write a computer code or statistical analysis, because we are dealing with large number of individual, like can be hundred of thousands of individual with million of genetic markers. So it's dealing with large data. So you cannot analyze it in Excel and SPSS. So it need to be like kind of special package. I think R is still okay. Or yeah, so it means that interested in numbers, have a computer ability, and then the rest is just studying understanding the basic genetic or something like that. Yeah.
Okay, thank you, Dr. Beben, for your answers. There is one participant who want to ask. Let's give the stage to Madam Yati. Bu Yati.
Yeah, thank you,
Please.
Pak Iqbal. Dr. Beben, thank you very much for your great lecture. It's very nice, yeah, to understand that the precision medicine is very give promise medicine platform, yeah. I just want to know the implementation in the Australia, maybe how this platform have been implemented there, because I imagine that the improve of the outcome and the reduce of the adverse effect from the precision medicine itself it's very great, but the implementation is, I think it's very cost. Yeah, the cost is the problem, because start from the diagnosing the disease, and choosing the medicine also, it's very cost. Yeah.
Thank you, Bu Yati, for the interesting question. So in Australia, probably similar also, or similar or some difference with other developed countries in Europe and then also USA. So there are several things how this genomic thing applied in practice or clinic. So first on single gene disorder or Mendelian genetic or penyakit genetik, there are number of genetic tests is commonly applied in the clinic. For example, in disease that I'm working on, like motor neuron disease, for example, motor neuron disease, about 10% of motor neuron disease has Mendelian form, where it's due to single mutation. So patients are regularly tested for what type of mutation they have before any probably as part of the diagnosis. And then also that's for a single gene disorder. I think there are also other number of gene panel that use commonly in the clinic. So there are a number of different gene for single gene mutation. For complex disease, at the moment, I think it's still quite early in the stage development where people trying to a clinical trial on how best use this genomic information, because it's not as in the sense that it is not as conclusive that okay, so if you have this mutation you get a disease. So, this is like more like kind of probabilistic, because there are a lot of gene involved, but it's still being trial, for example, on how to incorporate this polygenic risk score in clinic. But there are also a commercial, I think also in on Indonesia as well, like the commercial company that probably you heard about 23andMe, and then other genetic company where you can send your saliva sample for $100, and then they'll come back with a prediction of like, okay, so with your genotype you are more likely to develop some disease, and then you have also probably single mutation in this, and then tell you about the ancestry as well, like that you are like partly a European or something like that, or some, and then, but in terms of using it in clinic, still in the early stage, but the promise is there. I think it's quite a lot of research being done in this area. Yeah.
Okay, thank you. Does this medication covered by the government insurance?
For some of the single disease, my understanding is that there are some of them that are covered by Medicare, which is government, Australian government BPJS. That's called Medicare. Yeah. Yeah.
Okay, thank you, Dr. Beben. Thank you, Pak Iqbal.
Thank you.
Thank you, Bu Yati. Thank you, Dr. Beben, for your answer. Sir, what is the limitation of pharmacogenomic implementation? What is the limitation of pharmacogenomic implementation in context with the precision medicine?
So the limitation is there are a number of limitations. I think for I need to check again, but for in some instance that before anyone given a medication, they will be tested for a genetic mutation, like for the one that very adverse reaction. I'll check again what the example, but there are some medication where before you're taking that medication, you need to check your genotype first. Otherwise, because we don't want you to have an adverse reaction, which is can be quite significant. So the limitation, it's costly. Yeah, I think, but probably now the cost is going down considerably. So I don't think cost is a limitation anymore. So I think the limitation at the moment with PRS is because it is still new. And then we still don't know how to incorporate base of this information, whether it is used as similar with any blood test, for example, diagnostic tool, like for example, the level of cholesterol, level of iron, or level of other biochemical markers, for example. So that's what we still don't understand. And then the other one is that because it's genetic information, that it has more ethical concern as well. Because when you got genetic information, that it is not only your information, but it is also the information of your relative as well.
Yes.
Because we know that relative will share the genetic information with you. It means that we will be able, if you got your information, and then that information can be used to be able to identify your relative or family. Yeah, I think that the second thing. That the other thing is also related to the insurance as well. That which is still a quite interesting topic. That some insurance might be, let's say you don't have the disease yet at the moment, but then you do the genetic testing, and then the genetic testing identify that you are more likely to develop this disease, this disease, and so on. Then genetic insurance company might charge you very expensive insurance, or might not want to insure you because they know that you are likely to develop disease, for example, because like insurance work in kind of probabilistic thing that they just so if that you have the information, then it might limit your insurance choice. I think that's one of the limited. The other thing, the limitation is because it's new as well. So because when come up to implementation, then it's mean that it's require some level of understanding of the genetic within the health practitioner. Doctor need to understand how to interpret the probability of the developing disease, and the nurse and all other, and then which at the moment still limited, but there are a lot of education, I think, and then also program from the government to make the genetic information or genetic education is part of the curriculum, for example, including the pharmacy as well. I think we were going to run like a genetic course for pharmacists here at the university, I think, but the time was not enough participants, so we need to postpone that. But the idea is that because pharmacy also involved in dispensing the medicine, and then if understanding that if some medicine will have a different reaction for people who have the certain genotype or genetic, then it can be important. Yeah.
Okay. Yes, thank you, sir, for your answer. There is another question from the participant from the chat column from Sifa Fauzia. She want to ask you: is pharmacogenomic cost effective? How can the cost of pharmacogenomic testing balance with the potential benefit of patients?
Yeah, I think in theory, it should be cost benefit, isn't it? Like I mean, like it should provide a good cost benefit analysis, but I don't know the specific calculation today. But I mean, in theory, for example, that rather than giving the same medication for everyone, where probably 20% will have no effect, and then 5% will have adverse reaction. It means that that's a waste of resources, and then also create another problem for some individual. That's probably not good. But if we be able to identify or only give medication to people who might benefit, then it can be cost effective. Yeah. Because you only give like 80% of the medication, and then change medication for the other 20%. Yeah. So, but I need to check again, like what the cost-effective analysis, but probably will depend on medication by medication or disease by disease. I think in terms of the cost effectiveness, yeah.
Okay. So we need to check whether based on the disease to disease or medicine to medicine to get the summary of cost effectiveness
Yeah.
of the pharmacogenomic. Okay, so thank you for your answer. That is the answer for Sifa Fauzia. Okay, there is another question, sir, from Buyati.
Okay. Yeah.
In the chat column, does the nutrigenomic has been implemented widely in Australia as well?
Yeah, so nutrigenomic, so there are several, I think it's a interpretation of it is what is nutrigenomic anyway. So it seems that in Indonesia it's quite a big thing, and I got this question quite a lot. So what your understanding of nutrigenomic here? Because it can be two things, Bu Yati.
Okay. I mean, I have already tested nutrigenomic already, yeah, in a private laboratory here,
Yeah.
and the information is quite a lot, yeah.
Yeah.
Actually, now currently I'm fighting with the obese status, yeah. So I just want to have a lot of information, including the genetic, yeah, the genetic. I just want to know what kind of food or what kind of food should I I have, or should I didn't eat, yeah, to help me to reduce the body weight.
Okay, based on the is it probably polygenic risk score, isn't it? So you send your saliva or blood, and then they tested
From
for
it's blood testing, yeah,
blood
blood testing that the information about the caffeine, the folic acid, and the some vitamins, and including vitamin D, and
so they tested the basically the genetic or the single nucleotide. Yes, so it is not as much common in Indonesia, and then the science, I think I might be biased, but it's still not well established. What I mean is a scientific. I mean, we know, of course, there are some mutation that influence how you metabolize food. For example, the lactose gene. Yeah, so it's a lactase gene that for some people who has that mutation or does not have mutation, people will not be able to metabolize the milk, or people who have some adverse reaction to alcohol. For example, there's mutation as well. And then but for most of the food on nutrition, there is no clear answer, in the sense that the effect size probably is not as big as that one. So that's why we consider that nutrigenomic application in terms of the choose the best food, like it's not scientifically well established at the moment, and then people not I think it's from the health perspective, there's not much in terms of research. Yeah, it's not as big as the pharmacogenomics. So yeah, because like in general, I think our understanding at the moment, it's healthy food are healthy for everyone, regardless of the genetic composition. The only thing that different by genetic composition is probably specific nutrition, specific food like milk, probably caffeine, or alcohol that people can respond. But for all other, I don't see much of the very robust scientific evidence that people probably should avoid apple or instead of eating banana or something. So it's not grounded based on very solid research finding at the moment. That's my understanding. So it's not big thing as well here in Australia. Yeah.
Okay, thank you.
Thank you.
Okay, thank you, Buyati and Dr. Beben for your answer. Actually, we are approaching the end of the Q&A session, but we still open the question for maybe one two more questions. There is one participant raise her hand. So please to Rahmi Triwahyuni to deliver your question to Dr. Beben.
Okay, thank you, Pak Iqbal, for this opportunity. So Professor Beben, thank you so much for your presentation. My name is Rahmi. I am one of the students in program Magister program. Now I'm in the second semester. Back then when I was in the first semester, I used to learn about the pharmacogenomic. So it's quite interesting for me to learn about it from you. Hearing those questions from Pak Iqbal and Bu Yati, it's quite similar. But what I wanted to learn about the Australian citizenship and also about this Australian health government is, you know, we still have no, specific clue about the pharmacogenomic itself, especially in Indonesia.
Yes.
So what I wanted to know about the pharmacogenomic implementation in Australia would be, how to implement the genetic information if the patient have to move to another hospital. You know what I mean? Is there any system that built by the health government from Australia to linkage to the genetic information from one patient to another patients if they move to different hospital from A hospital to B hospital? I mean, how do they manage the genetic information to implement the medications itself for the patients? I think that's the first questions.
Thanks, Rahmi. So yes, that's another good question. So first is the medical, just my limited understanding about the health system in Australia. So it's based on the personal experience. So medical record, usually we have the main access point, I think, in Australia is we deal with clinic or GP or dokter umum. Yeah. So it's all of the medical record information are kept with a GP, with the clinic. And then, for example, if we need referral or go to specialists, yeah, so one of the specialists, whether it is here is a dermatologist or other things. So it means that and then so it's starting with GP. So GP refer the patient based on their assessment that, okay, I think this condition need to be referred to specialist. And then if specialist and then accept us, so we make appointment with specialist and then we do the test, for example, some any blood test and everything. Usually the record is copied as well to the GP. So it means that in addition to specialists, as the result of the blood test, the GP will have the result of your blood test as well, because like a GP will know all of your condition, not only the particular conditions, because like people can have a multiple different condition, for example. So, and then also the government level, so they have my health data where, but people is not compulsory to subscribe to my health. It's basically that my health data is for individual patient information that are kept in GP can be transferred to information across Australian health system, which you can say yes or you can say no. Because I think why some individuals say no, for example, because they are concerned about the security of this data, privacy, and so on. But I think to say yes, that probably it makes it easier for other treating specialists in different location in Australia to access your health information as well. So yeah, that two system. I think genetic information are also treated similarly. My understanding is with the kind of other clinical information. So they need to be keep in secure location, and then the access are private, and then you only able to, or doctor able to share them with other specialists is under if you give them consent, consent that you agree to share them. Yeah. Is that answering your question? No?
Yeah. Talking about the pharmacogenomic, if someone wanted to, get the consultation from one doctor,
Yeah.
They need to do the retest, or just once to do the, genetic information, or
Yeah,
is there
so
a limitation for the report?
yeah, there is no need for duplication because the record will be kept. So I mentioned that similar with genetic information, similar treated with other clinical information as well. So it's mean that all of the base record or everything you do with your health is kept within the GP clinic, general practitioner. So it's mean that if some other practitioner would like to have that genetic information that you have done be used with other specialists, for example, then you give consent to give that information to the clinician. So you don't need to take them again because that is a waste of resources, because the information will be the same anyway. Yeah. So it's all
Okay.
yeah. It's
Thank you
there's
so much.
some sharing, but there's some restriction as well. Whether it depends on the yeah.
Thank you, Prof.
Yeah. Thanks.
Okay, thank you, Prof. Beben and Rahmi for the question. This is the last question, sir. The
Yeah.
last question because how pharmacogenomic is studied in Australia, especially in higher education, and more specifically in pharmacy department.
Oh, at the university, you mean?
Yes.
Oh, yeah. So that's a good question. I don't teach the pharmacy student, but I have a colleague who pharmacy lecturer department. My understanding is very limited. It's very limited teaching on pharmacogenomic here within the pharmacy department. It's so that's why we were planning to have that kind of a course, I think, for pharmacy, I think. But they have, I think there are some curriculum on the genetic, but it's quite limited, I think. It's not, yeah. My understanding, I need to check again with my colleague how but yeah.
Okay, thank you, Dr. Beben. I think this is the end of the Q&A session. Okay, thank you to Dr. Beben for your answers, and to all participants who already active during the Q&A session. Sir, is it okay if we still have questions to ask you through your email?
Yeah, that's fine.
This?
Yeah.
Oh,
Yeah.
okay.
Sure. Yeah.
Okay, thank you, sir. Okay, the end of Q&A session mark the end of this lecture. I, on behalf of all participants, would like to thank Dr. Beben for his willingness to deliver his knowledge on the role of pharmacogenomics in precision health. And I, on behalf of the committee, would also like to thank all participants who were already active during the Q&A session. Okay, Dr. Beben, the committee has something to present to you as appreciation.
Thank you.
Okay, this is the certificate.
Thank you.
Please accept this appreciative certificate, sir.
Thank you very much, Dr. Iqbal, for the nice certificate here. Thank you.
Okay, you're welcome, sir. That is all for today's presentation. Once again, thanks to all of you. Good afternoon and best wishes. Next, I return this event to the Master of Ceremony.
Okay, thank you so much to Associate Professor Beben Benjamin, our today's amazing speaker, and also Mr. Iqbal, our today's amazing moderator. It was such a great and insightful discussion.
Thank you very much again for everyone for listening to my presentation today, and then also asking a lot of good questions. Yes, I'm happy to answer more through your email, through my email also. And then thank you, and then I'll see you again sometime. And thanks for Agnes and Dr. Iqbal, and then Professor Saripudin, yeah, as a Dean of Faculty of Pharmacy. Thank you.
Right, thank you, Professor Beben.
Thank you.
With that, this marks the end of our event. On behalf of the committees, we would like to express our sincere gratitude to our speaker, moderator, and all of the participants who joined us today for this international guest lecture. We apologize for any shortcomings that may have occurred during this event. Once again, thank you so much for your attention. I'm Agnes Anindita, today's Master of Ceremony, signing off. Wassalamualaikum warahmatullahi wabarakatuh. Shalom, Om Santi Santi Om, Namo Buddhaya, Salam Kebajikan, Salam Pancasila.